Have you ever been bitten by a mosquito before? After being bitten, did you experience a high fever? If so, then you have likely been infected by the dengue virus. The dengue virus, or DENV, is a common virus transmitted by mosquitoes. Every year, there are about 400 million dengue cases, and recently, there were about 195,000 cases in the Philippines. Despite its widespread prevalence, there is no well-known treatment that targets the virus itself. This is because dengue has 4 serotypes, or 4 variations that have different surface proteins. This is where we come in. Good day, we are Katrina Dela Rama,
Precious Siazon,
and Corel
from Philippine Science High School main campus, presenting our research on the in silico screening of 40 antiviral phytochemicals as inhibitors of the envelope protein of dengue virus serotype 2. Plants contain naturally occurring compounds that may have medicinal properties called phytochemicals. 40 phytochemicals with proven antiviral properties were tested for their effectiveness in inhibiting the envelope protein of the DENV serotype 2, which is the variation of DNV associated with more severe dengue symptoms. The envelope, or E protein, is responsible for viral entry into a cell. Thus, if it is inhibited, the virus cannot spread to other cells. Additionally, we assess the oral bioavailability of these phytochemicals, which is the ability for a drug to be absorbed by the body and take effect. This assessment was done using Lipinski's rule of 5.
To better understand how these phytochemicals may inhibit the E protein, think of a phone as the envelope protein and its charger as the human host cell. If the host cell interacts with the charging port or the active site of the protein, then the function of the protein activates. However, if we can find another structure that can also interact with the charging port or active site of the protein, then the protein can't interact with the human cell and activate its functions. Using molecular docking software, we found that 7 phytochemicals created strong bonds with the E protein's active site, passing our binding affinity threshold of negative 7.3 kcal per mole. Binding affinity describes the strength of the bond between the phytochemical and the E protein, with the more negative binding affinity indicating that the phytochemical is less likely to break off. However, only one phytochemical passed the criteria set by Lipinski's rule of 5. Glabridin.
Glabridin has a binding affinity of negative 7.6 kcal per mole and interacts with the amino acid residues of the E protein active site, effectively preventing DENV from spreading to other cells. It can be extracted from licorice roots and possibly be synthesized into a drug that may be used to treat dengue fever.
Again, this has been Katrina Dela Rama,
Precious Siazon,
And Corel.
Here are our references, and thank you for watching.